Hey, well, welcome back to the Classics in Immunology Journal Club. Today we're going to do a real classic paper. And if you like this video, like the video and click on the bell, and then also scroll down and on my to get to my website and all the other things with social media so that you can see what's out there. Yeah, all of the classic papers of immunology are on my website. And so if you read all these things, you don't have to read anything else in immunology. And so today we're talking about a paper that came out in Nature in 1975 by George Kohler and Cesar Milstein. And this is a very famous paper because this is the paper that described how to make monoclonal antibodies. And of course, in the days of COVID now, we've been talking about monoclonal antibody therapies, and monoclonal antibodies are now pharmaceutically, one of the major players within the pharmacopoeia of all the major drug companies. And of course, that's been now 35 years or so, 40 years. It was first described in this paper that we're going to review today in 1975. And it's entitled The Continuous Cultures of Fused Cells Secreting Antibodies of Predefined Specificity. Now, to give you some background on these two individuals, Cesar Milstein, who was the mentor, and George Kohler was the postdoc. So it's an interesting story from the standpoint of trainees, particularly. Milstein was born in 1927, and he died in 2002. And he was born in Argentina. His parents were from the Eastern Europe, and had immigrated to Argentina. And he ultimately went to the University of Buenos Aires in Argentina and got his bachelor's, and then worked towards a PhD in enzymology. And and then soon thereafter, he won a scholarship at Cambridge, 1958. So he was about 30, 31, and he went to Cambridge and he did a second PhD in Cambridge. In Cambridge, they would throw out their PhDs like you only had to study a year or two to get a PhD out of Cambridge. Piece of cake, So anyway, he studied enzymology again in Cambridge, and while he was there, he met Fred Sanger. Fred Sanger is notorious for the fact that he has won 2 Nobel Prizes, and he had just won his first when Milstein was there in Cambridge. He didn't work with Fred Sanger, but right at that particular point in time. But then, after he was done with his second PhD, he went back to Buenos Aires and was given a nice post within the University of Buenos Aires. But there were problems, political problems in Argentina. And in '63, only a couple of years later, he said, "I'm out of here." He went back to Cambridge because Sanger had given him an open invitation to come back and join. At the same time, Sanger had collected a bunch of other people, having been a Nobel Prize winner. Everybody wanted to come and work for him and so forth. So it was quite the place to be. So he went back to Cambridge, and Sanger told him that a hot item in immunology was to try to figure out how the body made so many different antibodies against a single particular antigen. I remember I talked about Baruch Ben Esaraf and trying to figure out the smallest common denominator of what would constitute an antigen. And so when they worked it down, it didn't have to be a very big molecule, but then the body would make thousands and thousands of antibodies to a single particular antigen, different antibodies, all reactive to the same antigen. We call that the polyclonal immune response, which is very, very good because it's very difficult to get around. It's like a huge multi-specialty army or navy that attacks the antigen. But the problem was that that's great for the immune response, but it's hard to study the antibodies and to try to figure out how the body genetically could come about making so many different antibodies to the same antigen. And at the time, it was known that multiple myeloma is a cancer of the plasma cell, and it had been described over the decades and centuries in humans. And what would happen is a terrible disease to get. What would happen is that the multiple myeloma means many bone tumors. because the bone marrow is where most of the plasma cells reside, cranking out antibodies. And so if one of them becomes, if only one gets a mutation that makes it grow autonomously, that becomes a cancer, and that becomes multiple myeloma. Yeah, and the bone marrow fills up with multiple nodules of cancer cells growing and making antibodies. So they make huge amounts of antibody. Well, people establish cell lines from those, and in order to try to understand how the antibodies were formed and so forth, there was a technique that they got into the lab in Cambridge called somatic cell hybridization. So what Milstein did was he took two different myeloma cells that made different antibodies. And of course, the problem with using that to try to understand how antibodies are made is that they didn't know what the antigen was for these antibodies. They were, because it was a cancer cell. He made somatic cell hybrids between two different multiple myeloma cell lines, one from the rat and one from the mouse. And there was a guy at the NIH who made his career in making more and more mouse multiple myelomas cell lines that people could study and so forth, and that's what they were doing. And the story then becomes interesting here, particularly if you happen to be a grad student or a postdoc, because Niels Jerne, who we talked about earlier, of the Jerne plaque assay in 1963, where he showed essentially that you could use soft agar and put sheep red blood cells on them, and then you could put another layer of soft agar with lymphocytes, splenocytes, and if an individual cell is making an antibody to the sheep red blood cells because they've been immunized and so forth, it would cause an area of lysis around the cell. And that was really the first demonstration of that Sir McFarlane Burnett's hypothesis of clonal selection theory was actually true, that a single cell would make a single antibody and so forth. Anyway, Jerne had just been made the head of the Basel Institute around it started in 1969, but they had to build a building in Basel, Switzerland, which is right on the Rhine River Valley, where it comes down from Germany and then takes almost a right angle. And it took them a while to build the building and so forth. They didn't really get going until 71, 72. And that's when George Kohler, who was a grad student in Freiburg, Germany, which is only, I don't know, about an hour's drive north into Germany. He was sent to Basel to be a grad student with Fritz Melchers, who was also German, in the institute. And Niels Jerne was the director. And Jerne collected all kinds of people, and they were really interested in this whole question of antibody diversity. How could you create such a diverse, and huge number of antibodies? So Jerne invited Cesar Milstein from Cambridge to come down and talk about his studies, because he was in the same sort of thing. And so the story I've been told, essentially, by other people from the Basel Institute or friends of mine, is that George Kohler was sitting in the audience, and Milstein showed his data on making this rat myeloma, mouse myeloma hybrid, and showed that they were both kinds of antibodies were in the fused hybrid. What you do, and what they had worked out, was you could fuse the cells so that you get a double cell, essentially. They call that a hybridoma. And in order to fuse the cells, you stuck in some Sendai virus. After Sendai, Japan, which is where the virus was originally isolated, it had a way to merge the membranes, the virus itself. They're tricky little devils, as we know. They do all kinds of things to us. But they took advantage of that, and they used Sendai virus to fuse these cells. And basically, That whole process was not, that was an art form at this particular point in time. You had to, it was difficult to do. Anyway, George Kohler, sitting there as a grad student and listening to this hybridization and the somatic cell business, said to himself, well, if you, instead of doing two different myelomas, if you took a myeloma cell that would grow autonomously because it was a cancer cell, and fuse that with a B cell from an immunized mouse that was making an antibody, maybe you could get it then, you could take advantage of the autonomous proliferation of the myeloma cell that would then make the antibody of predetermined specificity, which is in the title of this paper. And it worked, which is a good thing. And as they say, the rest is history, because they went on to win the Nobel Prize in 1984, 9 years later, after the publication of this paper. And it was the generation of a whole new era in immunology, because now everybody wanted to make monoclonal antibodies to their favorite antigen so that they could then use the antibodies for whatever purpose they wanted to. And the pharmaceutical industry thought, well, this is good because now we can make antibodies that of predetermined specificity against whatever, and we can use them therapeutically. Now, the only problem with that is that it took a long time, and I'll get in maybe at the end of this, I can re-talk about that. So in the introduction of this paper, and this is not another one of these major papers, the letter, it's only 3 or 4 pages long. They make several points. The first one is that it was possible to do, well, the question was, could you make hybrids between multiple myeloma cells and immunize splenocytes in order to make monoclonal antibodies? Would that work? And of course, that was George Kohler's idea. And what happened back at the Basel Institute is when he got his doctorate, he asked Yerny if he could go to Cesar Milstein in Cambridge and become a postdoc there and learn this somatic cell hybridization. Now, the importance of that whole thing is that Yerny's plaque assay, so Kohler learned the Yerny plaque assay. He knew he could probably do that in his sleep, you know? So he goes to Cambridge and the rest is history. So I'm sorry, I digress a little bit. The other thing that they wanted to learn about was this diversity question in terms of these antibodies. And they wanted to know whether or not if you fused these two cells together that had one cell making one kind of antibody and another cell making another kind of antibody, whether the different parts of the antibody molecules would resort, and you'd get all kinds of things and so forth. And so that's one of the things that they wanted to look at. And but they knew already that the so-called heavy chains and the so-called light chains from one antibody, a molecule didn't switch over when they did these rat mouse hybrids. And so they wanted to repeat that in the mouse, essentially. So that's the background. Now, in the materials and methods, they selected 2 different mouse myeloma cell lines that made 2 different kinds of antibodies. One made an IgG1, and another one made an one made an IgG2A isotype. These are isotypes, and they were both sensitive to selective medium, so that you could put chemicals in that would kill off those cells unless they got genes from a fused hybrid. So the 2 parent cell lines would get killed off, and only the hybrids would grow. This is good. And that was the experimental way you made these somatic cell hybrids. So in order so they could make these hybrids, so they made these hybrids, and Kohler's contribution was to try this with immunized spleen cells. Evidently, other people had done it. It was very difficult. It wasn't working, and so it just, you could do the 2 myeloma cell lines, but when you tried to use normal cells with the myeloma cells, they were having difficulty making that work. After they made the hybridomas, they were able to clone these things, and because they could put them in soft agar and a very low cell density, and so that you could see you could get little colonies growing up and so forth. And what they could do then, they could then pick those colonies and they could grow them in suspension individually and study them, which is what they did. And in order to make antibodies that they could define biochemically, they could put in radioisotope precursors of amino acids into the cultures, and they would become incorporated into the antibody molecules. And since the antibody molecules were the major product of these cells, they could then examine the immunoglobulin molecules by STS page, polyacrylamide gels, or isoelectric focusing, and biochemically then look at the different chains that these cells were producing. So, in the results of the first successful experiment, when they fuse these cells together, 4 out of 10 plates, petri dishes that they plated the things out in, grew in the selective growth medium that would kill the 2 parents. This is good because they got 4 out of 10. That wasn't bad. When they wanted to find out whether or not these cells that were growing were really hybrids, and so they did karyotypes on them. They did they looked at the chromosomes, and they just simply counted them, and they found that the hybridomas had about twice the number of chromosomes from either parent. So the normal valve seed spleen cells had 40. And the other ones had around 60, 65, and the combined had about 100, and that they put that into table 1. I'm not going to show you the data from this paper because most of the data are biochemical and in gels that are very difficult to discern, actually. You have to read it yourself to convince yourself whether or not what they say in the text is actually what they demonstrated in their figures. When they examined the antibodies that were being produced by their hybridomas, and they would subject the antibodies to reduction of the disulfide bonds, which they did by putting 2 mercaptoethanol in, they could see that the hybrids had extra bands on there that were not present in either of the parent, and so how they interpreted that is that there were new immunoglobulin molecules produced by mixes between the heavy and light chains. This is before reduction and then after reduction between the heavy and light chains of these different antibodies. And that answered one of their questions, is they knew there were variable chain segments of the immunoglobulin chains and constant regions. And they interpreted by saying to themselves, well, that means that's already present. It's not happening in the cytoplasm. It had to happen at the genetic level. And that meant that there were 2 different segments of chromosomes that were somehow coming together to make the different 2 chains. So then what they did was they, in order to find out whether or not the cells were making antibodies to sheep red blood cells, which is what they were trying to do in the first place, they would put the hybridoma cells into a layer of soft agar in a petri dish, and then on top they would layer the sheep red blood cells plus complement to see if they could lyse the sheep red blood cells. George Kohler tells this great story that he did this over the Christmas holidays, this experiment, the very first experiment, and he took his wife to the lab with him because he was so afraid that it wasn't going to work, and he thought that they could go out drinking afterwards or something like that, and say whatever. But it worked, and he saw all kinds of lysis of the sheep red blood cells, and that was the end of the whole thing. And so in the discussion in this Nature paper, they say, well, their results in the very first paragraph, they say their results showed that this is going to be a very powerful tool to produce specific monoclonal antibodies. And they said that you could probably use, they conjectured, for the myeloma cell is not one that made its own antibody, but rather there was a mutation that so that it couldn't make its own antibody, but it was still from antibody producing cell, and you could fuse that with the spleen cells, with the immunized B cell, and you could then you get just the immunized B cell product. And they thought, because based on their numbers and how efficient everything had worked, that it must be that the immunized cells with spleens, with the activated B cells, those activated B cells, blast B cells, fused better, more easily than non-activated B cells. And they finished by saying that you can use industrial techniques and so forth to mass produce antibodies. And the interesting thing is, at Cambridge, where they were, this was sort of this was in the dark ages. This was before what I've told you about the Bayh-Dole Act that was passed in 1980 in the United States. And so at Cambridge, they did not patent this whole thing. And Kohler himself, and then also Milstein, were very, very fundamental, nerdy, basic scientists. And they didn't want to have anything to do with this industrialized stuff, although they recognized that it could be used for that, and they thought that was a good idea. But they didn't patent it, and they didn't make bajillions of dollars and so forth. But then they went on to win the Nobel Prize 9 years later, and they got some money from that and a lot of notoriety. People have said that if you win the Nobel Prize, that's worth about $100 million. So, it's like being an athlete. You can charge all kinds of money for your endorsements and stuff. So I can't remember what else I was going to tell you, but I'll tell you next week. So tune in next week. And don't forget to like this video and click on the bell and check out the papers and read this particular one. So thanks so much. It was fun.
So after they gave the Nobel Prize in 1984 to Kohler and Milstein, what happened next was that Kohler was offered a new position to go back to Freiburg University. He was in Basel at the Institute, and to go back to Freiburg and direct a Max Planck Institute, which he did in 1985, and where he remained from 1985 to 1995, when he died of a congenital heart problem and congestive heart failure at the age of 48. But between 85 and 95, he decided that the action in the business was really into cytokines and cytokine receptors. And he had a paper out in 93 showing creating the first interleukin-4 knockout mouse, and then the next year, the first interleukin-6 knockout mouse. And so he really continued to focus on the B cell compartment, trying to understand how these B cells were being helped by the T cells to produce their antibodies. So that's it for this journal club, and it's been great, and thank you very much.